Hello, everyone, and welcome to the ReProgram podcast. I'm your host, Dr. George Murphy. Today, we're delving into one of the most fascinating and perhaps the most mysterious topics in science and medicine, aging, and the bold question of whether it can be slowed, stopped, or even reversed. Now, before we begin, let's acknowledge something important. Unfortunately, just like the Matrix, no one really knows what aging is. Textbooks would define aging as the time-related deterioration of the physiological functions necessary for survival and fertility. That's a mouthful. So complicated to say the least. And yes, we can describe its really obvious signs: wrinkles on the skin, gray hair, joints that ache, organs that falter. In the lab, we can actually measure potential markers of aging, things like cellular damage, DNA methylation changes, and maybe differences in telomere length. But if you ask 10 scientists for a definition of aging, you're likely to get 10 different answers. Is it programmed by our biology, perhaps? Is it just the gradual accumulation of wear and tear? Or is it something else entirely? The truth is, aging is still a mystery at its core. And yet, for as long as humans have been telling stories, we've been obsessed with escaping it. Ancient myths gave us the fountain of youth, water said to restore vitality and grant eternal life. Alchemists sought their elixir of immortality. In Greek mythology, Eos begged Zeus to make her mortal lover immortal, only to discover that he aged endlessly without death's release. Across cultures, aging has been treated not just as a biological process, but as a profound existential puzzle, something to conquer, cheat, or at least try to understand. Fast forward to today, and that mythical quest has migrated into the lab. Scientists are exploring things like senolytic drugs to clear away aged cells, gene therapies to perhaps reprogram biology, stem cells to regenerate tissue, and even interventions that seem to reset biological aging clocks, at least in animals. There aren't just ideas that are existing on the fringe. These are ideas that are serious efforts with serious funding behind them. But it's important to keep all of these expectations grounded. Age reversal is not just a pill away. We don't yet know if it's possible in humans to reverse aging or augment it in any way in the ways that we dream about. What we know is that the frontier of aging research is shifting rapidly, that's for certain, and it's forcing us to rethink what it may mean to grow old. So today, we'll explore where science stands, the myths, the hopes, and the very real breakthroughs that are starting to blur that line between science fiction and medical reality. So let's get started. And remember, knowledge is power. So first and foremost, what is aging? Many experts view aging simply as damage accumulation. Perhaps a simpler way to think about it is that for every biological or physiological action that happens in your body, there are products, byproducts, or shrapnel that's produced that's damaging, and that this damage builds up over time. So in our laboratory, we study something called dynamic resilience, or the ability to respond to stress or recover from disease or disability. So in this way, one might define aging as simply the loss of this resilience, or perhaps it's the loss of that fine balance between the insults we experience throughout life and this robust resiliency response. So perhaps a way to start to think about aging would be via something called the Hallmarks of Aging. So this is a nice framework for understanding aging, and the so-called Hallmarks of Aging are sort of a list of what might be considered to be these underlying drivers of aging. And these are things like maybe genomic instability and or errors that occur in your genetic code that could lead to disease or disarray or disorder. Other things are senescence, or the creation of zombie-like cells in the body that aren't really alive or dead, but may produce factors that cause damage or in and of themselves promote aging. Another one is mitochondrial dysfunction, or decrease in the functionality of your mitochondria. If you remember from your high school biology, your mitochondria are the powerhouses of your cells, and as these become dysfunctional, maybe that's what drives aging. Another is stem cell exhaustion. We love stem cells, and perhaps aging arises when your residing pool of stem cells, which we know provide the basis for all the cells and tissues in your body, maybe when they become depleted, That's what leads to aging. And lastly, one that is particularly intriguing for us in the laboratory is something called the loss of protein processing control. So we have interrogators in our bodies that are activated in order to prevent the production of aberrant or bad proteins, like, say, those that cause neurodegeneration, like Alzheimer's disease. And as we age, these mechanisms begin to fail. So yes, this Hallmarks of Aging is a nice framework in which we can begin to think about the factors that drive aging, but it's really also quite limiting. It actually comes from an earlier adaptation of the hallmarks of cancer. And just like with cancer, it's almost impossible to fit topics as complex as aging and cancer into these defined boxes. But I will conclude by saying, lastly, one of the other hallmarks of aging is something that's a little bit more complicated called epigenetic alteration or change. And this is a great jumping off point for our next topic. So obviously, to start, if we are going to study and eventually one day understand what aging is, we have to be able to accurately measure aging. So much so that if we were somehow, someday, able to augment this process, slow it down, or perhaps even reverse aging, could we monitor this and calculate the differences that actually occurred? So one of the ways in which we might be able to measure age is through the use of something called biological aging clocks. Although these are something you may have heard all over Instagram, real biological aging clocks do exist, and there really is significant science behind them. So your chronological age is your actual age as listed on your birth certificate. And your molecular or biological age, on the other hand, might be a more accurate predictor of your health status or longevity. So what we can do is we can compare your chronological age to your biological age, which we can generate through molecular data from you and compare these two independent ages. So in general, if you have a lower biological than chronological age, then that would be considered to be a good thing and may be related to your potential health span, longevity, etc. The conceptual but provocative thing here is perhaps your biological age is a potentially malleable variable, something that you can change, that can be augmented by things like diet, exercise, or maybe even one day a novel therapeutic. So one of the ways we can generate your molecular age data takes us back to that concept of epigenetic change. So what on earth are epigenetics? What is your epigenome? And how can we use this information to predict your biological age? The human genome was first sequenced over 25 years ago at a cost of roughly, amazingly, $3 billion. Today, we can do this overnight for about $200. So when the Human Genome Project wrapped up in roughly 2003, one of the biggest surprises wasn't just that we had finally mapped our DNA, it was the number of genes we found. So scientists had expected humans to have maybe 100,000 genes or more, enough to explain the complexity of all of our bodies and minds. Instead, we came in at around a paltry 20,000, only a little more than a roundworm. So what does that mean? It means the magic of being human isn't about how many genes we have, but rather how these genes are used, regulated, and orchestrated over time. So think of it less like the number of instruments in an orchestra and more about how well the conductor and the musicians can stay in sync. And here's where aging comes in. As we grow older, we don't simply lose genes. We really change the way in which our genes are switched on and off. The patterns of gene activity, what scientists call epigenetics, they start to drift. So the orchestra begins to lose its rhythm. Some instruments play too loudly, others too softly. And then the harmony that kept everything working smoothly, it starts to unravel. So this is why researchers talk about epigenetic clocks. So molecular patterns that can tell us our biological age more precisely than counting birthdays. So it's also why new approaches to age reversal are so exciting. Instead of just trying to fix one gene here and there, scientists are looking for ways to reset the entire system, whether that's through reprogramming cells, targeting longevity pathways like mTOR and sirtuins, or even clearing out senescent zombie cells that throw this orchestra off key. So the story of our 20,000 genes is really the story of aging itself. It's not about the notes on the page. It's more about how the music is played and whether we can learn to bring it back into tune. So to summarize, epigenetics and your epigenome, which literally means above the genome, are the master conductors of when and how our genes are expressed. And this gives us the wonderful complexity that's found in all living things. So how about a more practical visual example of how this occurs? All right. So I've got a Slinky here. Disclaimer, this is a massively simplified example, but let's pretend for a minute that this is your DNA. When I stretch it out, you can see how the coils separate. There are big open spaces, and it's really easy to reach in between the coils. In your cells, when DNA is in this open state, the cell's machinery can get in there and read all of the genes. Those genes can be expressed, turned on, modified, and basically used. Now watch as the Slinky collapses back down. The coils pack tightly together, and suddenly it's really hard to get in between them. The DNA is still there, but it's locked away. So the genes in that section can't easily be read or turned on. They're effectively silent. So aging, at least in part, is about changes in this pattern. Too many important regions getting locked down and others becoming open and noisy when they shouldn't be. The Slinky's the same, the DNA sequence is the same, but the way in which it's packaged and decorated changes with time, and that changes how our cells behave. I think an important point is that aging is the great unifier. No matter who you are, rich or poor, you can be certain that you will age. But a really interesting question is, does everyone age at the same rate? Certainly not. And even anecdotally, we know this isn't the case. It doesn't take a scientist to note that some people, at least by way of their phenotypic or functional characteristics, seem to age at different rates. We probably all know people at opposite ends of the spectrum. People who look freakishly young for their age. Others that seem to maintain functional capacity far later on into their life. So why do some people seem to age so much better than others? Genetics? Sure. Environment? Probably also a key factor. Resilience, as we've discussed? Yes. So all of these factors are current subjects of intense investigation. But the one thing that this tells us about the process of aging is that it's probably malleable, changeable, tunable, if you will. And in this way, perhaps even slowed down or stopped altogether. So our final and million-dollar question is, how can aging be augmented, slowed down, or maybe even reversed? Really interestingly, age reversal is actually possible, at least at the cellular level. So back to the Slinky. Here's the exciting part. Scientists are now learning how to reset the Slinky. And this is through things like epigenetic reprogramming, in which we're able to gently reopen sections of DNA that have been locked down with age, and maybe even close off ones that have become too noisy. So the idea isn't to change the Slinky itself. The DNA sequence stays the same, but to restore the pattern of openness and closure to something closer to youth. So in other words, age reversal at the molecular level might be less about rewriting our genetic code and more about teaching the Slinky to play the right tune again. So I hope you enjoyed this discussion. More to come on each of the subjects we broached. So please continue to follow our podcast and join me on this journey of understanding and self-discovery as we navigate our way into the fields of longevity, aging biology, regenerative medicine, and the emerging field of geroscience.